All right. Hello, everyone. Welcome. I think we can start the session now. So, my name is Hoon Cho. I'm one of the co-organizers of this session, along with Bonnie Berger, who is joining us remotely today. So the goal of this session is to introduce the ISMB community to recent progress in genomic privacy and security literature to increase awareness of this topic. And today we have an amazing set of speakers, including one keynote, 4 invited talks, as well as 1 abstract presentation. And there will be a wide range of themes related to genomic privacy. But first, to set the stage for our speakers, I'm going to be giving a brief overview of the field to introduce key concepts for those of you who may be unfamiliar with this field. So let's first talk about what we mean when we say genomic privacy and security. So privacy at a high level means having control over who has access to one's personal information and how this information is used. Security, on the other hand, refers to the quality of systems that protect against malicious attempts to access data or misuse it. And while these two concepts often go together, and often they're used interchangeably, it's good to know that they're different, and one does not necessarily imply the other. And we're generally interested in building systems that are both secure and protects the privacy of individuals. So why do we care about privacy in biomedical research specifically? So many of the data sets that we're used to working with in computational biology, they're in fact from real individuals. So we have, as researchers, we have this responsibility to minimize harm to study participants that can be caused by privacy intrusions. But it's not just about responsible research. Privacy issues can also be a key barrier to scientific progress by making it difficult or impossible to share data across different institutions and labs around the world. And this means that better strategies for protecting privacy can actually unlock new data sets that can give us new ways of getting at scientific discoveries. And similarly, translating new findings into the clinic and into the public health domain will require trustworthy systems that can act upon patient data while protecting their privacy. There are several unique aspects of genomic privacy that distinguish it from other types of personal information, such as those collected on the internet. So for example, genetic data reveals a lot of valuable information about one's health and behavior. And there's still a lot that we don't know about what can be inferred from one's genome. One can even make inferences about one's family or relatives. And genetic information is also static in that it stays mostly the same over one's lifespan. And lastly, the genetic sequence is also highly unique in terms of being an identifier of the person. So one of the earliest works on genetic privacy in 2004 showed that only 30 to 80 SNPs out of tens of millions that we're used to working with these days can be enough to uniquely identify a person. Now, almost 2 decades have passed since this work, and now we know a lot more about the myriad of ways in which genetic privacy can be breached in different contexts. The graphic on this slide shows a high-level classification of known attacks from a recent review article. And some of the seminal works represented here include Homer et al., which show that even aggregate-level summary statistics can breach privacy by revealing whether a person was in the dataset or not. There are also Genomic et al. There are also Genomic et al. And Ehrlich et al., both of which showed that one can use public genealogy databases to identify a person in a genomic sample. And these 2 latter works are actually from our keynote speaker, Yaniv Ehrlich, so hopefully we will hear more about these works in a few minutes. So despite the growing literature on this topic, we're still far from having a complete knowledge of genomic privacy risks. And part of this is because biomedicine is constantly going through innovations and experimental technologies that are generating new types of data. For example, recent advances in omics biology, including transcriptomics, proteomics, metagenomics, they all introduce new implications for genomic privacy that we're not yet fully grasping. But what is known is that many of these new data types reveal genotypic information, for example, through quantitative trait loci or QTLs, which can in turn be linked to the genetic profile of a person in another data set to reveal their identity. Our second speaker, Gamze Gursoy, will touch on the topic of omics data. An important approach to tackling these privacy issues is through policies and regulations. Here are some notable examples in the US and EU Note that this is an incredibly complex landscape, and different countries have their own different legal frameworks for this. In general, though, while these regulations provide important safeguards for the use of personal data, they are often ambiguous when it comes to genomic data, partially because our knowledge of what the risk factors are is incomplete. And updating these frameworks based on new developments in the field is naturally a very slow process. And I believe this is where privacy and security literature plays an important role by formalizing what we know about the privacy risks and also proposing new mechanisms for privacy that go beyond what is currently possible. So one line of work in this endeavor is leveraging what are called privacy enhancing technologies, or PETs. For those of you who are familiar, these include techniques like homomorphic encryption, differential privacy, and federated learning, all from the computer science literature. The main idea behind these methods is that computational methods can help us share sensitive data while also protecting privacy, thus helping us to resolve this seemingly inherent conflict between privacy and data sharing. There's a fast-growing literature on applying these techniques to biomedical applications, and I believe we're not far from seeing them being used in practice. And you will hear more about these techniques from some of our speakers, including William Yu and David Furlicher. Now, looking ahead, there's still much remaining work to do in terms of designing privacy-enhancing algorithms, especially in terms of addressing their scalability issues and analytic flexibility. And it's important to note that these methods won't solve everything. Working on social aspects of data governance, such as privacy regulations and informed consent structure, and so on, is just as important as developing these methods. And I believe our last two speakers, Bradley Mullen and Lucila Machado, will help us think about some of these social issues that come up in the broader context of their work. And finally, what is becoming increasingly clear is that going forward, we'll need more interdisciplinary efforts in this field. We need to combine the knowledge of what is needed in biomedical research with rigorous technical solutions for protecting privacy, as well as social frameworks and mechanisms that guide and complement the use of these technical solutions. And in my view, this audience at ISMB has a lot to offer in these areas. And I hope this session will inspire at least some of you to start engaging with some of these challenges. So with that, I will pass it over to Bonnie Berger to introduce our keynote speaker.